just to let you know, I had a YouTube link here, and it turns out that the author of it took it away. We gotta develop our own, get animation and do our own. But anyway, we'll find something for y'all in lecture and talk about this, because it is really good to see this in action. So again, we were talking about in the last video how we achieve the excitation contraction. Okay, so here, it's all going to be happening here in the beginning, right here, as that axon terminus comes down and releases neurotransmitter. In this case, it's always going to be acetylcholine. Okay, so these little pink dots are acetylcholine. It's always abbreviated ACH. Sorry, mine's lowercase, but anyway. All right, acetylcholine is the chemical communication. And acetylcholine, right, moves from an area where it's high, the axon, to an area where it's low, right, the sarcolemma. And it makes contact with a receptor. All right, so here's the acetylcholine receptor in the membrane. If the acetylcholine receptors are not there, nothing is going to happen. So everything is imperative. All these parts have to be there in the beginning in order for us to get the response we want. So acetylcholine receptors present. This acetylcholine will move quickly and make contact with them because they have a specific place for them to make contact. All right, when they do bind, so when acetylcholine, right, binds to its receptor on the sarcolemma, this makes a change in an associated structure, right, with the receptor. And this associated structure is a sodium channel. All right, so this is called, just so you let you know, a ligand-gated sodium channel. Because this is a ligand, this little acetylcholine. Not going to use that term on a test, but that's, again, there's different types of sodium channels to let you know. All right. So sodium, the other thing to understand is that sodium was very high outside of each of these cells in this area that we call the extracellular, outside the cell, and very low inside the cell, whether it's this muscle cell or this nerve cell, this part of this nerve cell. Okay. So because it is high outside and low inside, when this channel or gate opens, the sodium moves in, right? It is a positive charge, right? So I don't know why they have a negative here, but the book, figures sometimes that's a positively charged ion, right? And so binding of the acetylcholine, again, to its receptor causes, right, cause and effect, the sodium channels to open, okay? So there is more sodium, as said, outside the cell, and so therefore there's a charge. Before the sodium ever entered, there was a charge across here. All right. It turned out that because sodium's high out here and low in here, right, we had this positive, 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 positive outside and negative inside. We always measure with a probe inside, so we say negative 60 to negative 90. All right. So the outside to the inside of the cell was charged before this happened. That was our membrane potential. All right. Once I let acetylcholine go, it binds to its receptor, and I open this channel, and the positive starts coming in, right? You can see that this is going to change because now it was positive outside, negative inside. Well, now with positive coming in, the inside is going to become more positive. So when things have opposite charges, positive, negative, we say they are polarized. So with the change, right, bringing the positive in and equaling, right, the charge is starting to become more positive inside and it's positive outside, we call this depolarizing, right? And again, it means that the inside is becoming less negative than it was. Okay, so charge is flowing, right? Current can be established. So, right, I said once we depolarize the membrane, right, the sarcolemma, this change in electrical potential will actually open more, and we call these voltage-gated sodium channels. So, and these are spread away from this junction along, here's a close-up of the sarcolemma, along the sarcolemma. So we change it in one area where the axon is meeting it, right? We've changed the electrical potential, we've depolarized, and this will then start up a chain reaction, right, where sodium channels will begin to open along the rest of the sarcolemma. All right, so the sodium gates are opening along here. You don't see this, they haven't drawn that in, but you certainly could. And eventually we get to one of these openings into the cell, right? So this is the boundary between the cell and the cytoplasm. So into the cell called that T-tubule or transverse tubule. All right? So the action potential starts to move into the cell because of this. So traveling along, opening, opening, sodium, sodium, opening, opening, and starts to go in here. It starts opening, right, because there's still sodium gates along this membrane, okay? So this membrane is right next to this membrane. This is a membrane of the sarcoplasmic reticulum, and it's a little bit wider here than it is as it spreads out, so we do refer to this as, right, a cisternae, you know, as I talk about cisternae here. I'll point that out in the models. It's not really a big deal, but what is important to know is that here is a place within the cell, so we're inside the cell, in which this electrical change is carried into the cell, and it can have an effect on structures within the cell, not just at the membrane anymore. So when this depolarization, which is due to sodium channels opening and sodium coming into the cell, happens, this will lead to the membrane of the sarcoplasmic reticulum, right, changing its electrical potential, which will then have an effect of opening calcium channels. So calcium, again, before the stimulation by the neuron, before any of this happened, calcium was all pretty much kept inside these tubes. So it was not free. And it was therefore separated from the myosin and actin you see outside. But now, for the first time, if you will, right, calcium is able to leave the tube, and it's going out into the sarcoplasm, where the myofilaments are. So we're going to pick this up. All right, and again, it's important to know that there is more calcium inside the sarcoplasmic reticulum than out, and that calcium is the necessary ingredient, as we said in a former video, for contraction. All right, so, right, muscle fiber contraction, how do we get here? So start with that acetylcholine, always have to back up to where we started and kind of trace everything back again, reinforcing. Start with the acetylcholine, released from the axon into the synapse, moving across the synaptic cleft and binding to a receptor that's in the sarcolemma of the muscle. All right, as we zoom in here. All right, this opens a sodium channel here, which changes the charge. Right, so now we have depolarized, and that's the term we use, depolarize the membrane here at the synapse. This electrical change causes electrical change to continue. We said it's like a wave-like propagation. And so this allowing sodium in here triggers sodium channels over here to open, right, without the need of that acetylcholine. And so they begin opening and opening along the sarcolemma. The sarcolemma moves into, right, so we're inside the cell, into the sarcoplasm as a T-tubule, and we continue to see sodium channels opening. Well, this T-tubule is in close contact to the membrane of the sarcoplasmic reticulum, which holds the calcium, right? So once we change electricity here, it causes a change here. This change opens, as the arrows show you, the calcium channels, which calcium now starts to leave the sarcoplasmic reticulum, shown here. All right? So this allows the calcium to bind, for the first time again, if you will, to troponin. So troponin, that little myofilament we talked about associated with actin, shown here, right, the double-stranded pearls, right, has a binding site for calcium. But if calcium's not around, it's not bound, and it stays in place. When calcium is available, right, it can now bind to troponin, and troponin, right, will change its structure, which will move the tropomyosin. Why is this important? Because currently, tropomyosin had been covering, well, up to this point, tropomyosin had been covering a place on the actin where the myosin could bind. So without that calcium binding to the troponin, the tropomyosin will not allow, right, myosin to bind to actin. And if myosin can't bind to actin, you can't get a contraction. So all these events have led to just this thing right here, myosin head binding to an actin. All right? And again, we kind of zoom out here to show that if we do this, we can start shortening the sarcomere. And when the sarcomere starts to shorten, and here we have the pattern for the, right, those A bands and I bands and A bands and I bands, right, we'd get the whole muscle shortening. All right? So we then relax, right, the muscle by returning all this, resetting everything. So after we've done a contraction, we can begin to relax. So in order to relax, right, we've got to move that calcium back, all right? So the calcium has to come off the troponin, which allows the tropomyosin to come back, right, reposition over the myosin binding sites on actin. Yeah, I know it's a lot of words, you got to keep track, right? And therefore the myosin can't make contact with actin anymore. And here again, the cartoon is showing you this calcium being returned. Now, I say right here, you don't see it in this cartoon, mentioned here though, that you must have ATP around to do this relaxation. And when we look at the actual contraction, like we introduced on this slide, you also need ATP. We didn't emphasize it here, but so we're going to discover that we need this energy molecule in order either to contract muscle or to relax it. Okay? And I do say, as long as calcium is bound to troponin, right, and ATP is available, calcium ions are around, muscle will stay contracted. And you can keep skeletal muscle contracted for long periods of time if you think about like your posture, standing upright, sitting upright, all right? But eventually you're going to, right, stop the contraction and you're going to reset, right? You're going to relax that muscle. And so this all begins when the motor neuron really stops sending acetylcholine. So as long as your neurons are sending acetylcholine, you can maintain this, and as long as you have energy and calcium. All right? So I say when you start that to relax, you basically stop sending, right? You choose this through your frontal lobe. You stop sending acetylcholine, all right? Acetylcholine is actually destroyed. So you can either take the acetylcholine back up, right? Reuptake is what we call that. Or you can destroy it. But that powerful chemical is started this whole thing, so we have to get rid of it. So we have this enzyme in this area called acetylcholinesterase, and it basically destroys acetylcholine. And you recycle it and reuse it, but it's gone. That's the point. And once it's gone, then no more binding to a receptor, no more sodium channels open. So I say here again, that leads to the sarcolemma, right, this membrane, and the T-tubules, which are part of the sarcolemma, repolarizing. What does this mean? Well, basically it means the sodium goes back out, potassium channels open, and we'll talk about that, to reset, right? So we want to get back to this where we had more positive charge outside than inside, all right? So some potassium leaves, sodium gets pumped back out. We'll talk more about this in future slides, but we're trying to reestablish that polarity, positive outside, negative inside, so we call that repolarizing. Oh gosh, I misspelled that. Jeez Louise. You guys, thank God I don't take off for spelling, huh? So calcium is pumped back again, back to the sarcoplasmic reticulum, requires ATP, tropomyosin covers back up the actin, and myosin heads come off, and the muscle relaxes. So that's, there's a few more details we have to include in this, but that's really it in a nutshell. All right. So in the next couple slides, again, we're going to reinforce this, but we're going to look more at this, what we call this Crossbridge cycling and this sliding filament model of contraction. So we've given you some of the basic details, and you really do want to keep those straight, and you really want to report them as a sequence, because it is a cause and effect, as I said, a cascade of events, all right? So here again, now we see the whole, as opposed to being focused in on the myosin and actin, right? We see the whole sarcomere represented, which is nice. So we get this idea of, again, shortening of the sarcomere as they see this. So I always say that the walls are closing in, all right, basically when you do contraction, okay? And you see, again, these myosin heads here. So this is a relaxed muscle. This is a contracting muscle. With the myosin heads making contact, the tropomyosin is off these binding sites. Here on this model, relaxed muscle, no contact, and tropomyosin is blocking, right, the binding sites on the actin. Okay, so our contractile cycle begins when myosin and actin can make contact, and it will continue as long as these two can make contact. Right after they bind, the actin will slide past the myosin to shorten. Yes, there is a limit to this, and we'll see this. The limit really is the end of the Z line, right? So muscle can only shorten so much. So we do have defined limits here. What is the value of the calcium leaving the sarcoplasmic reticulum? Well, that again allows, right, the troponin to move the tropomyosin. What does it bind to? Again, just reinforcing that, it binds to the troponin, and this then moves the tropomyosin, and now myosin and actin can make contact.